I am going to talk about evolution, not really about statistical physics. This talk really ought to be in the day in evolution, but because of my annoying schedule, we're doing it now. But as we'll see, I'll talk a little bit about some of the theoretical aspects of this work, the theoretical population genetics, and that actually does have quite a lot of flavor of statistical physics. So what I'm going to do is talk a bit about both experimental and theoretical approaches to try to understand why natural variation looks the way that it does, and sort of to try to provide a foundation for understanding why genetic variation looks the way it does in nature, and also just how the evolutionary process picks and chooses among all of the stuff that could in principle happen in any given system to determine which mutations actually succeed in populations, how evolutionary change progresses, and why diversity looks the way it does. So I'm just going to give a little background to start with, because I think probably a lot of you guys have not seen a lot of work in evolution before. So one of the things that we see all over the place is, striking examples of evolutionary change. And so there are a number of really famous examples of this throughout the literature. Here, I'm going to just take you through a couple of examples from some of my colleagues. So here are Hopi Hoekstra's mice. And if you look at these mice, you'll see that there's natural variation in coat color. This natural variation in coat color, Hopi has shown through lots of really painstaking work, is adaptive in the way that you might imagine it's adaptive. On this sand, it's better to be this mouse than this mouse. On this sand, it's better to be this mouse than this mouse, basically because you get eaten less by birds. So there's a clear selective effect here. And this sort of evolution of coat color has happened repeatedly and rapidly as these mice have expanded or changed their range to different sorts of sand. Okay? Basically, whenever they sort of expand into some region with some different color sand, quickly these different types appear that match their coat color to the sand and presumably help avoid predation this way. These are lizards from South America, where there's a whole range of diversity of the shape of their snout. You can't see it here, but the weight and shape of their tails. Obviously, this sort of striking morphological differences here. This is perhaps the most famous example of Darwin's finches. You see a bunch of different skulls of different finches that live on the Galapagos, and they have different beak morphology, different head shape, and so on. And there's really a ton of work in evolutionary genetics which aims to understand and explain this phenotypic variation. And these evolutionary geneticists can now tell us a tremendous amount about why these evolutionary changes are adaptive. They can tell us, for instance, what the specific mutations are that underlie this diversity in beak morphologies. And they can explain, exactly how at least some of these mutations change the expression levels of certain transcription factors at particular stages in development in order to generate these different morphologies. So you can really say quite a lot about what has happened in evolution. But until fairly recently, this is really primarily an observational science. And sort of we're just explaining this kind of evolutionary history. And there wouldn't really be much for us to talk about at a meeting such as this. But nowadays, there's really quite a bit we can say quantitatively about exactly what's going on. So before I get into some of that, we can also see sort of in more detail about the evolution, about the signatures that evolution leaves in our genomes. So, in organisms such as humans, for instance, we obviously have lots of phenotypic variation, but because there's been such a tremendous amount of effort placed into sequencing our genomes, sequencing the genomes of many, many different individuals, we can actually say at a much finer level about variation within human populations. So here is one example from a paper from a few years ago, looking at the pattern of fixed differences between humans and chimpanzees over the chromosome 1. I mean, this is just one example. Obviously, they have the other chromosomes as well. This is across chromosome 1. This black line or gray line here is the divergence between humans and chimps as a function of position along the chromosome, and these red and these red and blue lines are covariating factors such as functional density across the genome. And you can see, okay, there's some complicated pattern here where on average the human-chimp divergence is a little less than 1% across this chromosome, but there's substantial variation here. And so we might ask, why does this pattern look the way that it does, and how can we build a framework for understanding that? Here's another sort of more recent data set just to give you an idea of the sorts of data that we're going to try to build a theoretical framework to understand. This is looking at about 6,700 human individuals, basically, sequencing their exomes. Basically, that's all the coding sequence in their genome. And in those 6,700 or so people, across these 6,700 or so exomes, there are a lot of different sites at which there are mutations in one or more of the individuals. And if we look at each of those sites at which there is a mutation, that mutation will be present in either 1, 2, 3, all the way up to at most all of the 6,700 sequenced individuals. And we can see most of those mutations, okay, are present only in a few individuals, okay. Almost like 10% of the mutations that they observe are present in only one or a very small, or 3 or 4 different individuals out of the 6,700. But there are many mutations also that are present in many more individuals, smaller fraction. Maybe 1/1000th of the mutations are present in half of the 67 mutations. And this has some characteristic form. This blue here is for African individuals within the sample. The red are mostly European individuals. So you can see there's a difference in the amount of diversity in the Europeans to the Africans. And so we might want to understand, to try to understand why why does this look the way that it does? Why are there these differences between Europeans and Africans? And what can we say about this? So this is the whole field of molecular evolution, is to try to explain this kind of genetic variation. So I'm going to talk primarily, though, today about looking at these questions in an experimental setting, because experimental evolution offers us a really nice tool to ask these questions in a much more controlled setting, where we can vary parameters and we can have experimental replication. It gives us a lot of useful tools. And so the kind of experiment that I'm going to be talking about basically involves putting bugs in flasks. Okay, so this is an example of this is a picture of the most famous evolution experiment done so far. These are 12 flasks that are maintained by Rich Lenski at Michigan State University, and he's been maintaining E. coli in these flasks for now 61,000 generations. Okay, he's been doing this since 1988, I believe, something like that, approaching 30 years at this point. And basically, he just puts some E. coli in the flask, lets them grow up overnight. The next day, he comes in, makes a new flask, and dilutes a little bit of the old flask into the new flask, and the E. coli just continue to grow and divide like this over now 26 years, something like that. Okay? And so what you can see in these experiments, that it doesn't really look as pretty as the finches or the lizards, but the E. coli get better at doing what they do in this flask. Okay, if you measure the relative fitness as a function of time, this is from a paper a year ago now, looking at this through the first 50,000 generations of evolution, the fitness of these E. coli increase. And I'll define what we mean by this fitness more precisely in a minute. But the point is that they do adapt. And like we can see in these kind of natural populations, like we just talked about with the humans, you can really very easily assay what the genetic variation is that has underlay this phenotypic change. Okay? So here is an example from a paper a few years ago again, where folks from Rich Lenski's group sequenced a single E. coli individual from generation 2,000, 5,000, 10,000, 15,000, and 20,000 from one of these evolving lines, from, which one is it? This one here. Okay, and they what you're seeing, each of these concentric circles are one of those genomes from each of these time points, early time points in the middle, late time points at the outside. And each of these little tick marks is a mutation that they observed. Okay? And how many of you know anything about E. coli? Okay, yeah, reasonable number. All right, so these are all of the genes, okay, in which these mutations occurred. And so you can see things like SPO-T, which is a strongly beneficial one, TOP-A, which they've studied a lot, PIKE-F, etc. Okay, there are a lot of these mutations, a lot of these things. If you know something about E. coli, you might sort of have some intuition about what these mutations might be doing. But the upshot is that using this kind of approach, you can systematically try to study which mutations happen, why they're beneficial, or in some cases they're not, what they're doing in any event, how they're accumulating through time, and so on. Okay. So just from this experiment, it's a little hard to say anything about why this happened as opposed to something else. So one of the sort of basic problems with studying evolution, in some ways it's like a little bit like cosmology. It's something that has happened once, and so if we look at things like this, okay, this is some signature of the single episode of human evolution. These are sort of single events that happened once, and at the same time, of course, evolution is a highly stochastic process, right? The mutations that occur are random. There are random effects. Somebody gets some good mutation but gets hit by lightning the next day, then it's not gonna get passed on. So there are all sorts of random effects that are important. And so, if we're going to say anything about this process, it's going to be some sort of statistical statements. And so another great thing about experimental evolution is that you can use replication to directly assay the statistics of different outcomes, right? So here is an example of an experiment conducted by Olivier Tenelian, who works here in Paris at INSERM, although he did this work in California, actually. So he took 115 different strains of E. coli. Actually, I take that back. He took 115 of the same strain of E. coli. Yeah?
Your previous slide, how do you define the fitness of the organism?
Yeah, I'm going to get to that in just a second. Just bear with me. At the moment, they're getting better, okay, in some ill-defined way. All right. So these guys, they took 115 lines of E. coli, put them in a high temperature, higher than E. coli likes to live in, for 2,000 generations. At the end of that time, and they started all from exactly the same E. coli, right? So they just took it, let it divide, founded a bunch of different lines from this identical genotype, and then 2,000 generations of evolution later, they sequenced a single clone isolated from each of these 115 populations, and this is the line, each horizontal line is one clone, and they've, depicted the mutations that occurred in each of these clones via these various colors, point mutations in red, for instance, larger insertions or deletions, larger deletions in purple, and so on. And so, you can see from this that there is this sort of complicated pattern of similarity and difference between these replicate lines, right? Some of these lines, many of these lines got some mutation at this location in the genome. Some smaller number of lines got mutations over here. There's some complicated pattern where some sites are sort of repeated targets of evolution, others are sort of less common. And somehow, we would like to be able to understand what this kind of statistics of evolution means about the underlying, the underlying process, okay? So basically what we are trying to ask in all of this stuff, everything that I've told you about before, is somehow about understanding what are all the mutations that could have occurred and their fitness effects and anything else about their biology. And how does that get transformed by this sort of dynamical process of evolution, okay, into what we actually see in these experiments or when we sequence natural populations. And so all of what I'm going to tell you about is going to be focused on understanding this here, okay? And I'll tell you about some experiments that we've done to observe these dynamics, and I'll tell you a bit about the sort of theoretical frameworks that we're working on to understand how these relatively simple rules, like how natural selection works, has been understood since Darwin. Okay, but still, it's difficult to predict, even if you knew everything that could happen, how selection will transform some set of possibilities into what you actually see. So that's what I'll be focusing on. Okay. So, it's a little further off what I mean by fitness than I had thought. Sorry about that, but we'll get there. Okay. So I'm going to focus a lot on thinking about adapting populations. Okay, this is certainly not the only thing we could consider. Obviously, a lot of evolution may be taking place in constant environments to which organisms are well adapted, and they're just sort of accumulating neutral or deleterious mutations. But at least for much of the talk, I'm going to talk about a situation where we imagine some population is adapting to some new selective pressure. Okay. And this actually seems to be very common, actually. Things are always adapting to something, it seems like. And so the traditional sort of view that most people have in mind when they think about how this process works is pretty simple, okay? The idea is that the limiting factor in how populations adapt is waiting for rare mutations to occur, right? The idea is somehow that mutations are some rare sort of precious thing. You have to wait for the beneficial mutation to happen, and then once the beneficial mutation happens, okay, here I'm drawing an example of a population, this beneficial mutation, which I'm going to call A, happens, and then it spreads through the population, and then before too long, everybody has it, and we call this a selective sweep. And then much later, maybe some other rare beneficial mutation happens, B, and that sweeps too, and so on. And this adaptation is this sort of succession of selective sweeps. And to the extent to which this is an accurate, intuitive picture of how evolution is happening, we can actually, people have long understood what sort of signatures this should leave in the kind of data that I've been showing you over the past few slides. Basically, each new mutation that happens, beneficial mutation that happens, quickly spreads through the population and sort of purges out all other diversity, right? Because you can imagine that if there were other mutations that had occurred in this time, neutral mutations or other things, this mutation B is just going to spread out and purge all of that diversity. And so there's just this succession of selective sweeps. We can understand that pretty easily. But what it seems like is that in really every population where we look closely, adaptation looks more like this. Okay, this is actually even an oversimplification. There's lots of different mutations happening in lots of different genetic backgrounds, and they're all spreading and competing and interfering with one another, and they can't all succeed at once. And so there's these sort of complicated interactions between all of these mutations where, in this example I've drawn mutation B winds up driving mutations A, D, E, and C to extinction. Okay? And this seems to be a sort of just generic phenomenon that happens throughout all sorts of adaptation. Okay? It is most sort of striking in an asexual population where these mutations can't recombine together, and I'm going to talk about experiments in that context, at least for the first part of the talk. Later I'll talk about recombination as well. But this generic phenomenon happens in any kind of organism, right? Because if you think about it, like, if you have kids, you are going to pass along whole big chunks of your genome to your kids, okay? And your significant other is going to do the same. There's going to be some amount of recombination, but there'll be whole big, many megabase chunks of your genome that you will pass on all of it or none of it. And so within those regions, if your wife has one of these mutations and you have one of these other mutations, or vice versa, then that one or the other has to succeed. You can't pass them both to your kids. Okay, and so this is why this sort of complicated dynamics is where as someone who originally was trained as a statistical physicist, I got interested in trying to understand these dynamics, and there's a lot of statistical physics tools that turn out to be useful. But before I get to any kind of theoretical thing, I want to just talk about the basic setup of the kinds of experiments that I'll be telling you about. I'm going to go sort of back and forth between experiments and theory through the course of the talk. And so I'm just going to start by setting up some of the experimental background here. So the experiments I'm going to tell you about are all done in budding yeast, just because that's what I happen to work in. And it's basically the same kind of experiments that I showed you about on the previous few slides. You take some yeast, you put them in a test tube, you let them grow up overnight typically, then you dilute them into a new sterile test tube, and you just keep doing this again and again. One of the great things about doing this in microbes is that, first of all, you can do this relatively quickly. We typically maintain about 10 generations a day. So that will give you a sense of the kind of timescales involved. You know, over several months, you can do 1,000 generations of laboratory evolution. You can freeze them, okay, which is extremely useful because then you can go back to them later. You can tune how you do this dilution to change the population size. You can also change other parameters like the mutation rate by starting with strains of yeast that have problems in their mismatch repair machinery. And now, as promised, the fitness here, in this kind of context, has a very simple operational definition. Basically, if you have a mutation which creates a new genotype, the fitness of that genotype is simply the average number of offspring that a single individual with that genotype will leave into the next dilution cycle, right? And so if that number is greater than 1, that will be a beneficial mutation. If that number is less than 1, that'll be a deleterious mutation. And this is a measurable thing. I'll talk about how we measure it on the next slide. In practice, we do these experiments. These are not test tubes. These are wells in 96-well microplates or 384-well microplates. So that lets us do thousands of these replicates using this robot. And what I'm going to tell you about, at least to begin with, and then later I'll tell you about more complicated things, but for the first part of the talk, I'm going to talk about haploid asexual yeast, okay? These are evolving in rich media, okay? Those of you who know yeast, this is why. How many of you have worked at all in yeast? Okay, okay. Essentially nobody. These are just maintained in YPD, which is rich laboratory media. And basically, this is the media in which these laboratory strains of yeast have been maintained for, since they were isolated originally from the wild. So for decades, they've been living here. It's the sort of simplest and dumbest thing that you could choose. And as we'll see, nevertheless, these yeasts are going to just constantly be adapting to this laboratory environment, despite the fact that they've seen it for long periods of time. And we're going to look at, these questions that I've been talking about, which mutations fix and why, and what determines the patterns of variation within these populations. So I'm going to talk a lot about fitness, and I just defined what I mean by that, and we can measure that actually quite precisely, okay, by just taking some evolved strain or genotype and mixing it with the fluorescently labeled reference strain, right? So you just mix these two, you maintain them exactly like you do the experiment, and then you measure the ratio at the beginning and at the end, right? And the difference in these ratios tells you exactly what you want to know, okay? Yeah? Yeah, so there's not really a great advantage for most of what I'm going to tell you about. I use yeast for historical reasons, essentially, associated with the fact that I was a physicist and knew nothing about biology and happened to get into yeast first. As it turns out, so originally I was thinking about yeast because we can maintain yeast, they're a eukaryote, right? You can maintain them both sexually and asexually and as diploids and haploids. As it turned out, for the first 10 years or so that I worked on yeast, we could not actually get them to have sex in the lab reliably. I'll talk about that later in the talk. It's actually a real pain in the ass to convince yeast to have sex. But we've been working on it for 10 years, and now we've got it working. So I'll tell you about that towards the end. So that was the original reason, but in these experiments I'll talk about for now, they could have been done in E. coli just as well. That being said, there's no disadvantage to doing them in yeast either. I mean, it's a sort of comparable system. In a way, it's sort of better because yeast genetics is like the best studied system, really. It's certainly convenient. But you could do these just as well in E. coli, really.
Is there a difference in how quick they grow?
Yeah, so there is, E. coli in principle could divide every 20 minutes, and yeast maybe about every 90 or so, 80 to 90 minutes at most. So it seems like that would be an advantage for E. coli. In practice, it's not unless you're really masochistic, because if you want to maintain a lot of these populations, you can't maintain them in a huge amount of volume because your lab is only so big. So if you want to maintain thousands of these things, you're talking about 100 microliter, at most 1 mil volumes. And so if you figure out, like, for the population sizes that you want to do, how often you would have to do the dilutions to maintain a given number of generations. If you wanted to do more than, so we do 10 generations a day, meaning 1 dilution every 12 hours or 1 dilution every 24 hours. We could do more with 90 generation doubling times just by doing, like, coming in and diluting every 8 hours, for example. But you can imagine that, like, convincing graduate students to come in every 8 hours for 6 months is not, it would not help with recruitment. And, you know, so to go really beyond that, like, you could try to do E. coli faster by coming in every 4 hours and diluting, you know, but, like, there's no way you could convince people to actually do that. So in practice, it doesn't matter. Okay. All right. Oh, the one thing I just want to say, so there's a lot of caveats with this, okay? This is a very simple way to measure fitness. In principle, you could have frequency-dependent fitnesses, for example. You could have non-transitive fitness interactions where, for instance, one of the evolved strains is more fit than this guy and the other is less fit than this guy, but the guy that's less fit, you know, is more fit in competition with this one or something because, you know, there's some specific warfare between specific strains of yeast and so on. So all of these things could in principle happen. In the experiments I'm going to tell you about, they don't. Okay? Fitness is a very simple sort of directional thing. Okay? Which is not to say that there aren't all kinds of interesting questions you can ask about situations where that's not true, but this is sort of the simplest possible setting we can think about how selection works. We do have a few specific examples where, for example, you get frequency-dependent selection, and that could be a whole other talk, but I'm not going to give that. Okay, so I'll start by just giving you some pictures of how these populations evolve. Okay, so over the course of 1,000 generations of evolution, they increase in fitness. Okay, these error bars reflect variation across different replicate lines. So you can see that, okay, there's a lot of variation, that, you know, there's just inherent randomness in the evolution, but they all do increase in fitness over the course of this evolution. So they are adapting, like I was saying, we were studying adaptation. And so then we can try to understand what the genetic basis of this adaptation is using just a lot of whole genome sequencing. So I'll tell you about some data from an experiment that we published about a year ago now, and then we'll talk about theory, how to understand this. Where here in this data set, what we did is we took 40 of these evolving populations, and at 12 time points indicated by these arrows, we just ground up the whole population and sequenced the whole thing. And so the idea is, like, if you have some mutation at some location in the genome, it's at 40% of the population at this time point. So you grind it up and you sequence the whole thing, and 40% of your sequence data will say mutation there, 60% will say ancestral, and you'll figure out, okay, the mutation was at 40%. And then you can track it through time just by looking at how that frequency changes. Okay. There is a whole complicated bioinformatics problem here, which I'm not going to get into, other than to acknowledge Dan Rice for doing really some complicated stuff to figure out how to handle sequencing errors. The key point is that we identify mutations that reach at least 10% frequency at some point, and then you can track them through the rest of the evolution. So this is what you see. Okay, these are 40 populations. Okay, 1,000 generations each, frequencies. Each of these lines represents a mutation that occurred spontaneously during the evolution of that population, and what happens to its frequency as a function of time. I'm just going to zoom in because you can't really see much there. Here are 8 populations where you can see in a little bit more detail. And so, I mean, the first thing that you can see from this is just that, you know, this scenario that I was talking about schematically is in fact going on. There's just a ton of mutations in these populations. They're, increasing and decreasing, and some of them wind up fixing in the population, meaning everyone, they succeed, everyone has them at the end. Others of them maybe go up in frequency. Here, these mutations then go extinct eventually because they're outcompeted by this other group of mutations. So you have all this complicated dynamics. And we want to understand, what can we say about how all of these complicated dynamics determine which mutations succeed and which go extinct and what the probabilities of those events are. All right. So the first thing we can see about all this is just looking across all of these populations. Okay? Mutations accumulate steadily through time. So they just appear, and then they go extinct sort of steadily through time, so you get some steady state polymorphism. So there's some sort of, across all the populations, some steady process going on average. But if we look back at this data, what you can see is there's a tremendous amount of sort of variation between lines, where specifically, in each line, the evolution is very punctuated. There's a whole bunch of mutations that come up, here like a half a dozen different mutations all sort of appear at once and sweep through. Here's a group of 5 mutations that increase and then decrease all together. And so there are these what we call sort of cohorts of mutations. And so, yeah.
I must have missed something, but what are the traits that you're looking for? What are the individual curves here?
So each of these curves is a mutation that arose at some point during the evolution. Some single nucleotide polymorphism typically, although some of them are insertions or deletions. Most of these are single nucleotide polymorphisms. I haven't really told you, I don't know if you can see from there, but there's some dotted lines and solid lines here. The solid lines are non-synonymous mutations in coding regions. The dotted lines, if they have a circle, they're a synonymous mutation, or if they have a square, they're an intergenic mutation somewhere. So you can see there are lots of those as well, although the majority of these mutations are non-synonymous mutations in coding regions. I'll talk a little bit in a minute about what these mutations are, but I haven't got there yet. Okay.
Excuse me.
Yeah.
Earlier I missed something of it. You just showed that you can mix the strains prior to sequencing. So when you did your sequencing, how did you know that the mutation came from which of the strains?
Oh, I'm sorry. I didn't mean to imply that. There are 40 populations sequenced through time. Each of them was done independently. What was mixed is the whole population was ground up. This wasn't like often people will take out a single clone, single cell essentially, and sequence that. Here we sequence the sort of whole population sample. And so the disadvantage of that is you don't have any single complete genome sequence of any individual. But the advantage is that you can sample some of the variation within the population. So if 10% of the individuals within the population have a mutation, you can see that. Right? And so here, that's what we're seeing, the frequency within individual populations of each of these mutations.
My question is really stupid.
Yeah.
When you say you sequence the whole population, you mean a representative part?
Yes, of course. Yeah. Yeah. Well, all I mean is we didn't pick clones. Yeah. It's an unbiased sort of sample of the population. Okay. I'm not going to really get into this, but the point is that this clustering, it's not just something that you're seeing by eye. They're really like groups of mutations that come up together, much more than you would expect by chance. Okay, and yeah. So by just looking at one of these populations, we can sort of get a sense of all the sorts of things that are going on to determine which mutations succeed. Okay? So here's just one example I'm going to take you through just in a little bit of detail so that you can see all the sort of factors. So this is the data. Okay? It's just one of the populations I showed you on the previous slide. I've just recolored these things just to guide the eye, basically. And then here I've drawn a schematic of this evolution in terms of this kind of diagram that I showed when I was talking about these issues of many things happening at once earlier on. So here what we see is this mutation in SPC3. Okay, this is a non-synonymous mutation in this gene. Okay, it occurs and starts spreading through the population. And there's in some other genetic background, this mutation in this putative ORF occurs and starts to grow. And then this group of 6 different mutations, okay, all indicated in black here, come up in some third genetic background. And it looks like these guys are going to win and drive these two to extinction, except this additional group of 3 mutations occurs here in this background that has this SBC31 mutation, and then this other mutation in We2 happens, and this group of mutations winds up outcompeting these other 2. Okay. In parentheses here, after each of these mutations, I'm showing the number of replicate populations out of the 40 in which we saw mutations in that gene. So this ROT2, for instance, or this UR1, or this We2, these are sort of parallel targets of evolution. They're certainly strongly beneficial mutations. And so what you can see from this is it's sort of not enough to be a beneficial mutation. You have to also sort of interact the right way, get lucky in the right way, in occurring in the right genetic background. I mean, just to give you a particularly striking example of that, in this population, there are 2 different mutations in the same gene, We2, that happened independently. This one happened in this wild type background, this one happened in this background, and this one outcompeted and drove this one to extinction because of these sort of stochastic effects of the interactions with other. Yeah.
How do you know that the purple subpopulation is outside of the light blue one, or it's an assumption?
Yeah, so I've sort of glossed over something, and I was sort of half expecting somebody to ask this question. So yeah, you're paying attention. I don't actually know that directly based on the sequencing data. However, if you think about the whole time course, this is the only way it can be, right? This cannot be on this genetic background, okay? Because it doesn't fix, right? If it was on this genetic background, it would have to fix. Okay, because these mutations happen later. And then, it has to be on this different genetic background because it goes down as these guys go up. And, if you just think about the rules of logic. So in a bunch of these cases, we've also gone and taken individual clones and genotyped them just to confirm this intuition. So here we actually know this for a fact. But in general, it's sort of obvious if you look at it carefully. Yeah?
Yeah, the purple one you say couldn't occur on the sign background, but it could actually have occurred on the sign background.
In the what? On this background? Yeah, that's true, actually. You're right. That's true. But that is not actually what happened here. But yes, you are. How do you know? Because we sequence clones from this population. But you're right. There's some ambiguity on things like that. It is a good point. I shouldn't be so.
Sorry, maybe I missed. There is a highly improbable that 6 mutations occur at the same moment.
Yeah, I know.
What happened at this moment?
We don't know. So first of all, they didn't happen at the same moment, presumably, because keep in mind we're only seeing these mutations when they reach at least, say, about 10% frequency. So they could have happened in relatively quick succession, and because each of them is beneficial, the sex-tuple mutant outcompetes the 5-tuple, outcompetes the 4-tuple mutation, etc., before they reach the threshold of detectability. Okay. That being said, if you try to simulate this process and try to ask, with theory whether that explanation that I just told you makes any sort of quantitative sense, the answer is no. It doesn't really hold together, so I can make that explanation, but it's really kind of bullshit. At least we don't really believe it. And so to some extent, you can get some clustering the way I just described, but 6 mutations at once like this is really hard to explain. And so we just don't know. There are a couple of possible explanations. I don't really want to get into. There's one which we've got some really nice sort of tempting evidence for now. One thing we were thinking of is transient fluctuations in mutation rate, which we have no evidence for whatsoever. Another thing is although I said there's no frequency dependence in these populations, that's true for anything that reaches substantial frequency. Maybe there are lineages that get 1 or 2 mutations and then are just maintained at very low frequencies until they accumulate enough mutations to break out. We actually have some evidence for that now, but I'm not going to show it. Yeah.
Are these genes especially close together?
No, they're scattered all across the genome. In these cases, I don't remember exactly in this particular case, but typically they're all totally on different chromosomes even. So it's not that it's like a single event.
Yeah.
Yeah. Yeah. No, like these guys are all on different chromosomes even. So it's, yeah.
And is there a difference in the rate of mutations whether it's introns or exons? Or?
No, it appears that this, there's no evidence for like clustering of mutations. With the exception of at the gene level there is. Okay, I'll tell you about that in a second. Okay, that's because of selection though, presumably, at least in large part. But no, like genome-wide, there's plenty of intergenic mutations roughly in proportion to this, the relative proportion of coding and non-coding sequence. At least, roughly speaking. So I mentioned I would talk just briefly about what these mutations are. I don't want to like dwell too much on all these experimental details because I also want to talk about the theory and so on, and where there's at least a little bit of math involved. But these are a bunch of genes in which there were 3 or more replicate populations got a mutation. And so, for the 1 or 2 of you that have seen yeast before, you can see what all these genes are doing. I don't really want to get into it. Sort of interesting thing, though, is that, even these genes, which are strongly beneficial and are, in this case, mutated in more than half of our replicate populations, they still fix only a relatively, maybe at best half the time in which they are observed, right? So it's not enough to provide a benefit by yourself. All of these kinds of interactions are also important. This is just another way of looking at that same thing, summarizing all the data, right? Out of 1,000 total mutations across all 40 populations, about a quarter of them fix. And you can parse that out into the different types of mutations. And the general question we want to ask is why are these percentages the way they are? How do we build a framework for understanding this stochastic process of evolution when there's a lot of things going on? Okay, so I'm obviously like not going to talk about all the details of the theory, but I just want to give you a flavor of how we try to model this kind of thing. So the idea is we have some complicated thing of what's going on, all these different mutations, okay, that are present. And what we are observing, okay, is the fate of individual mutations. And just to sort of, so we're on the same page of what we're actually watching, right? If we look at this mutation, okay, that occurred here, its frequency through time would be like this, because it, increased additional mutations happen in this genetic background. That we don't see in this trajectory itself. But we see there's sort of indirect effects in maintaining this guy, maybe drawing it up, before these other mutations in other genetic backgrounds drive it to extinction. And this is what we observe, but this is sort of underlying. And here's an example of this mutation, which is one of the ones that helped out this one, but didn't wind up succeeding and went extinct. And here's another maybe somewhat more successful mutation, okay? And we want to model these kinds of dynamics. So there are really 2 components of this that we look at. So basically the difficulty here, okay, as you can imagine, let me just make sure. The difficulty with doing this is that obviously stochastic effects are very important. You have to have some kind of properly stochastic model that accounts for the fact that these mutations are random and the genetic backgrounds they occur in are random. At the same time, there are nonlinear interactions between the mutations because, in the end, there can only be one, right? That's not really quite true. But in the end, like all the mutations that are on different genetic backgrounds, only one of them can succeed. And so, there's some nonlinear competition there between them. And so that combination of the nonlinearities and the stochasticity is difficult to handle. And so what we do is basically divide up the 2 things. Okay? On the one hand, we sort of develop some sort of mean field-like description of the overall variation in the population, which properly accounts for the nonlinearities. And basically the idea here is you have some distribution of fitness within the population at any given time. Some individuals are more fit than others because these individuals have better mutations than these ones, okay? And there's some variation in that, okay, in the population. And that variation is maintained because new mutations are constantly arising while selection is destroying the less fit variants. And so there's some balance between this process, okay, whereby you create this sort of steady state variation in fitness, which of course as the population evolves, it's moving towards high fitness, but with some roughly average, at least average shape, okay? Which we can calculate what this average shape, what the shape should look like on average based on the mutation rate and the population size and the fitness effects involved, okay? And then, from the point of view of the fate of each individual mutation, we treat this overall variation as fixed, okay? And then later, we'll self-consistently demand that all of the fates of individual mutations are consistent with what we've assumed about the overall variation within the population, okay? So the idea here with each individual mutation is pretty simple. There's all the other stuff going on in the population has generated this blue curve describing the variation in fitness within the population. And each new mutation, it occurs on some genetic background with some fitness in proportion to the frequency of that fitness within the population. So here I've drawn an example where a beneficial mutation occurs in this genetic background, which is more fit than average, okay, but is not the fittest background in the population. And it confers some advantage. I'm just assuming this is a mutation that confers this advantage S individual. And it then jumps out front, okay, because it has a benefit and it's occurring on this background that already is more fit than average, creating a lineage that is, this much more fit, S total more fit than the average, okay? And then it increases in frequency because, I've drawn this example being more fit than the average, so it increases in frequency, okay? Looking at time going back this way. And in the meantime, the rest of the population starts to catch up. This blue curve moves to the right and it, starts to catch up. And then, as time goes on, if nothing else happens, no additional mutations happen in this red lineage, the rest of the population will catch up and pass it by and this guy will go extinct, okay? Now, alternatively, of course, we could imagine additional mutations happen in the red lineage, sort of allowing it to surf out in front, and that's how it would fix, right? So this process is sort of also observable. Okay, it's just another way of looking at these trajectories. So this is like a schematic of one of these trajectories. And here we can imagine this initial rate of increase in frequency of that particular lineage is a measure of how much more fit you got than the mean, right, at the beginning. Because that's basically how quickly you rose up is based on how far ahead you were of everyone else. Okay? And this is a measurable quantity, okay? In these experiments. Well, I'm going to call it the landing fitness because it's sort of where this mutation landed. And so basically, if you think about it, all of this question about which mutations succeed and which fail, how repeatable the evolution will be, is about this tension, okay, between the fact that on the one hand, there are some presumably very strongly beneficial mutations, okay, but there are only a few of them. By definition, there's only a few best things that you can do. So things like IRA1 or ROT2 or WE2, these genes that were mutated in many replicate populations, presumably are very strongly beneficial, but there are only a few of them. They can land way out front here if they occur on, essentially any genetic background, okay, because they provide such a strong fitness advantage by themselves. But of course, they only happen rarely. On the other hand, there are presumably lots and lots of neutral or even deleterious mutations, but they can only land way out here in front if they happen to occur in exceptionally lucky genetic backgrounds, okay? So it's this tension between how rare are the rare mutations versus the common mutations versus how lucky are these backgrounds, how rare are these lucky backgrounds compared to these backgrounds that determines the balance between these mutations fixing versus these mutations fixing. Okay? And that's what's going to determine the statistics of all these outcomes that I've been talking to you about. And so we have all these papers that describe the quantitative details of this, but that's the basic idea. And so you can see signatures of this in experiments. So I'll just tell you, in these experiments, just tell you a couple of examples. So here is that landing fitness as measured in the way that I told you about from these experiments, and the fixation probability. And you see the further ahead that you land, the more likely you are to succeed. But that given that you land at a given place, it doesn't matter. The mutations that are intergenic or synonymous or non-synonymous all have about the same probability of fixation. Okay, in other words, a bad mutation on a good background is as likely to fix as a good mutation on a bad genetic background. Same, you know, as I was just saying. Okay, you see a sort of complicated effect of population size. All right. On the one hand, smaller populations, they have less mutations are occurring. So those, you know, strongly beneficial mutations are less likely to happen in the first place, right? Because there are just less individuals in which the mutations can occur, so you sort of less thoroughly sample the full mutational space. On the other hand, when a good mutation does happen, there's less other stuff going on, and so there's less competition. So given that the mutation, once it happens, the good mutations are more likely to succeed. And so you can see that here, given that you land a given distance ahead of the rest, you're actually more likely to succeed in a small than in a large population.
I'm sorry?
Yeah.
Fitness, could you redefine it because I'm not sure I've got
Fitness.
And how do you measure it?
Fitness here is defined as, on average, how many more or less offspring do you leave in each new dilution cycle relative to the average individual within the population at that time. Okay? And you can measure that by using fluorescently labeled strains, okay, which you mix in with some strain you want to measure the fitness of, and you measure how many more or less offspring one or the other type does by counting cells, basically. Okay? And you can actually measure that rather precisely. I mean, we can do like measure thousands of strains in parallel to like 0.3% accuracy in this number of offspring that are generated.
Actually, I
Yeah.
have a question about that. So, when you have, for example, when you want to compare fitness, do you compare it with the same fluorescently labeled
Yeah.
cell? So
Right. So you know, say you have 100 strains and you want to measure the fitness of each of them relative to each other, but you can't do that directly. You measure each of them relative to the same fluorescent strain, right? And then, you know, you just do the subtraction, basically. And so in principle, I was getting at this earlier, but I don't know if it was clear then. In principle, it could be the case that there are non-transitive interactions, such that I'm not allowed to just do that, okay? And that does rarely occur, but you know, we've done a lot of sort of surveys with multiple fluorescently labeled strains just to check for this kind of thing, and you know, it essentially almost never happens. So in principle, that could be a complication, but in practice, it's not. Yeah.
If I'm correct on this, your definition of fitness impinges on your ability to grow faster. But in some backgrounds, this may not be the fit criteria.
So it is a complicated thing if you think about mechanistically. It's growing faster would certainly make you more fit, but actually, like if you look at these mutations, a lot of them are also, for example, in knocking out stress response or in knocking out negative regulation of RAS. And what those things are basically is, I mean, the short answer is what those things do is they prevent the yeast from stopping growing towards the end of a cycle quite as much. So they may not divide any faster, but they divide for longer even when the densities get higher. And so, you know, yeast, historically, it has paid for them to be pessimistic. Like usually in the natural history of yeast, you know, they want to sort of notice when things are getting bad, because when things start to get bad, they're likely to get worse. Okay? I mean, that's like, you know, the tragedy of yeast in the wild. But in these happy laboratory conditions, right, you know, we call it they're learning to trust us. You know, the graduate student will come and the media will get better. And so getting bad does not mean that you're about to get worse. And so there's things associated with that, for instance. Anyway, all this is sort of an aside. It doesn't actually matter for what I'm talking about. But the point is that there's some complicated sets of many different sort of underlying phenotypes that, but you know, in this particular environment, they combine in some specific way to define this number of offspring that you leave, okay? And that's a well-defined thing. It depends on some complicated way on all these underlying phenotypes like growth rate and trusting us and et cetera. But I mean, we're interested in that, but I'm not going to talk about that anymore, yeah. All right. Another nice thing, all right, I'm going to move on to like a little bit. I have until 1:15, right?
yeah.
Okay. So, you know, a nice thing about yeast is that you can cross them and map things. So, you know, I've been talking a lot about fitness effects of individual mutations, and I've kind of been talking about it by inference. But it's nice to know that in these yeast, you can actually take evolved strains, cross them to their ancestors, genotype the offspring, you know, isolate offspring with individual mutations or specific combinations of mutations, measure their fitness, and by that sort of directly map the fitness effects of individual mutations. And so, you know, we've done that in some of these populations, and it makes sense in terms of what I've talked about before. I don't want to get into it more than that. Because I want to sort of shift gears and talk about a slightly different question now. So, so far I've been talking about, you know, what is it, how do we understand which mutations fix and which don't, and, you know, how all these interactions and stochastic effects determine that. And that's sort of relevant for thinking about, you know, if, you know, you adapt for a long time, all you're going to see at the end of the day is the mutations that fix. And so, you know, that's sort of relevant for thinking about how selection shapes, you know, which mutations underlie divergence between species. But I also showed data like right at the beginning, for instance, where we were looking at, you know, variation within species. Okay, like when we think about variation within human populations, we're looking at variants that haven't fixed. You know, many of these mutations that are present, you know, in this room right now, most of them will go extinct. Okay, sort of bad luck for that for you, if you have one of those. Anyway, but that's the sorry fate. And the other sort of thing is, in these experiments, we've had the advantage, because it's an experiment, of having a whole time course, right? We were looking through time, and we were using the whole temporal dynamics to try to figure some things out. But of course, when you go and you sequence lots of natural populations, you often just can't do that. You just have mutations from, you have individuals from one moment in time, now, and you can sequence them, and that's all you've got. And so, one of the things that I've been working on a lot with Alex and with others, and we've been looking at experimentally as well, is what happens, you know, how do we understand what this kind of variation looks like when we have sequences of individuals from a single time point, okay? And so, you know, I'll tell you a little bit about the experiments, a little bit about the theory. This is a figure, okay, where we just sort of, to mimic this situation, we threw away all of this beautiful time course data that we had from these populations. And we just said, well, what if this was a natural population? And we took one of our yeast populations. Okay, we actually took a bunch of them. This is one I'm showing you. And we took, I think it's 24 different individual clones, okay, from the final time point, and we sequenced them. So this is like what you would do if you were just sequencing some natural population where you didn't have access to the time course. And then you can plot the genetic variation among those individuals as a phylogeny, right? This is the genetic relationships. All the non-synonymous mutations in coding genes are listed here. There's a bunch of other synonymous mutations and intergenic mutations as well. And so you can just see the pattern of variation between the individuals here that you got. And so the question is, you know, if this is all the data that you had, and that is the case in, you know, sequencing human or many other natural populations, how could you tell that there was all this natural selection, all this adaptation going on? How much could you say about all the stuff we know is happening just based on this kind of snapshot of genetic variation at a given moment? So the way that people understand this typically, so this is of course a long-standing problem in population genetics, which people have been working on for 100 years. And the sort of traditional approach is to think genealogically, okay? So what you do is you imagine, okay, I'm sequencing a bunch of individuals from the present, and I want to think about their genealogical relationships, okay? And so using an approach called coalescent theory, we can compute the probability of a given family tree of these individuals, okay? So these are, I'm just drawing some putative family tree here of all these individuals in the present. As we look back into the past, they're descended from this common ancestor, and they, you know, had these relationships. And we can compute the probability of any particular genealogy, okay? And, you know, this sort of long-standing theory to do this, you know, we can compute this probability assuming that there is no selection going on, okay? And so for those of you who've heard about it, this is sort of like related to the neutral theory of biodiversity. The idea is if there is no selection whatsoever, you can compute the probability of any genealogy. And then you can imagine, okay, mutations happen at random, okay, on these genealogical branches. And, you know, I'm just drawing one schematic here of where mutations might have happened at random. And then if you think about it, that leads to this particular pattern of mutations on this particular pattern, this particular genealogy, leads to this particular set of sequences when you sequence, right? Like 5 of these individuals have this green mutation. That's these 5 individuals. Sorry, I'm running out of thing here. And, you know, 2 of them also have the red mutation. 1 of them has this yellow mutation, and so on, right? You can get from this genealogy to this data, okay? And so we know the probability of all this stuff, and so we can compute the probability of the data, okay? And so, you know, we can make various predictions about how we expect this data, some statistics describing this data to look like, okay? I don't really want to get into it, I guess, because I don't have that much time left. But the upshot is that we can predict things like, you know, what we might expect this frequency spectrum to look like, how much rarer we might expect common variants to look rather than rare variants, for example, just based on thinking about these statistics, okay? Now the problem is that when you go look at it, okay, this data actually looks totally different from this prediction, and that seems to be just generally true in lots of populations. You never have things that really look like this, okay, would predict. And that could be for a variety of reasons. And of course, you know, given what I've been talking about, I mean, you know, for example, in human populations, a lot of it presumably has to do with the complex demographic history of humans, right, coming out of Africa and spreading through the rest of the world. But here, what I'm going to talk about is how selection affects this, okay, because that's what we've been talking about for the past hour. And if you think about it, natural selection just sort of totally causes this whole approach to break down, because I've assumed that I can compute the probability of the genealogy and then imagine putting mutations at random on that genealogy. And that doesn't even make sense if we start thinking about selection, because the whole point of selection is that the mutations affect the genealogy, right? If this was a deleterious mutation, it should make this genealogy less likely, because why would this individuals with these deleterious mutations give rise to so many offspring in the present, right? So you can no longer have this decoupling between the genealogy and the mutations, and this whole approach breaks down. So with Alex and other folks, we've been working on how to extend this coalescent theory to describe this more complicated situation where selection is going on, to try to form a framework for interpreting this kind of data. And so I'm just going to try to give you, again, like an intuitive picture of how this works. Okay? The basic idea is pretty simple. You imagine, again, this distribution of fitness within the population, like we've had before. And you imagine sampling individuals. Let's say I sampled 3 individuals from the present, okay? And they just come at random from this distribution, totally at random, okay? And then let's think about their ancestry backwards in time, right? If we look a little bit in the past, okay, if you think about it, it is likely that the individuals in the present are descended from more fit individuals a little while in the past. And the reason for this is sort of intuitively obvious if you think about it, right? So, like, just think about this room, right? How many of you guys don't have any kids? Okay. So many of you don't have kids. Many of you are young, so there's still time for you. But some of you will never have kids, right? How many of your parents don't have kids? Right? Your parents all have kids, right? So some of you low fitness individuals who are not going to have any kids, right? Nevertheless, your parents were more fit than you, right? Obviously, some of you all are going to have many kids and whatever. So this is only true on average, of course. But on average, your parents are more fit than you. And your grandparents are more fit still, because not only did they have kids, but they had good kids who also had kids. And your great-grandparents were even better than that, and so on, right? So as we go backwards in time, this sample of individuals from the present is descended from an ever more and more fit subset of the population in the past. You know, in the past, there was presumably all kinds of variation in fitness as well. But it's only the most fit individuals in the ancestral population that left offspring into the present. I mean, that's what it means to be more fit, right? Fundamentally, right? And so if we think about it, you are as we go backwards in time, this sample is descended from a smaller and smaller subset of fitter and fitter individuals from those ancestral populations. And so it's as if the population size was declining in the past, even though it doesn't have, I mean, maybe it actually was, but even if it wasn't actually declining into the past, it's sort of as if it was, because you're only sampling ancestors from the more and more fit part of the population. And so we can, calculate, based on this fitness distribution and the statistics of the mutations, exactly how this process works and how that modifies the coalescent process. And the upshot of it is that it leads to this kind of skewed branching, okay, and these multiple merger events that change the expected patterns of variation in the data, okay? So here's an example of a genealogical tree from influenza rapidly adapting that one of our collaborators, Richard Nair, has been studying. And you see these kind of signatures. And these are our experiments, which at least sort of qualitatively have similar sorts of distortions. OK, so in the last just couple of minutes, so far I've been talking entirely about purely asexual populations. And I want to just say a few words about how what I've said is all not just completely an artifact of microbial populations that don't recombine much, but are actually relevant for all sorts of organisms. I mean, think about linkage and selection, how these things work now when we add recombination. And so just for those of you guys that don't know much biology, this is you here, okay? Half of you comes from your mommy and half comes from your daddy, okay? Here is your significant other. And this is the genome from your father-in-law and your mother-in-law, okay? When you guys have a magical event and produce a child, what will happen is there's going to be recombination in each of the chromosomes, typically of order 1 recombination event per chromosome arm, okay? In both you and in your spouse. And then, so this creates a kid that has some genome, which includes, on average, half from your mom, half from your dad on one copy, and half from your father-in-law and half from your mother-in-law on the other copy. This is, of course, only true on average, right? And so there are these huge blocks, is the point, which are inherited intact from, mother, let's just take from mother to kid, and then this part comes from grandma or grandpa with some probability and so on, right? And of course, the further you go back, the more these get chopped up by recombination. So the further in the past we go, the sort of shorter these inherited blocks are. And so this sort of gives us the idea to think about all of these things that I've talked about in terms of what I'll call a linkage block. Basically, the idea is that we all have this genome, okay? If we consider regions of the genome that are very far away, such that in the time since the common ancestor, there will have been lots of recombination events between these two sites that are distant in the genome, these are basically evolving effectively independently, okay? Because, one comes from one set of parents, the other comes from some totally different set, and they're basically evolving independently. On the other hand, on short distance scales, okay, where there isn't much time for recombination to occur, okay, there's not much probability of recombination, I should say, on the time scale since the common ancestor of some set of individuals, these are evolving effectively asexually. And so basically the idea is that we have these sort of linkage blocks. Short distance scales evolve according to all this stuff that I was been telling you about, and then different blocks evolve more or less independently, okay? So we can use all the stuff that I've been telling you about more or less out of the box to understand how, genomes of sexual populations evolve, provided that we can appropriately define what the linkage block is, right? And then different linkage blocks behave mostly independently. This is obviously a drastic oversimplification. There's lots of details here which we haven't completely figured out. There's still a lot of stuff to be done here, but this is the basic idea.
Excuse me?
Yeah.
What about the borders of the block?
Yeah, so that's like the stuff that's not worked out. Right? I don't actually mean that there is a specific border, right? These borders are entirely fuzzy, right? And in any pair of individuals, there will be specific borders that happen to be, you know, from whatever the particular history of those individuals are. And if you have a larger sample of more individuals, you know, there will be some set of breakpoints. You know, this is defined by where recombination has actually happened in the past. And, you know, we can only statistically try to characterize that, right? You know, what the distribution of block lengths is and what the distribution of where the recombination events have happened is. And, I mean, we don't really, this is like stuff that really needs to be worked out. We've just started to try to understand this. Okay, so just in the last couple of minutes, we've been trying to look at this. You know, I just talked about the theory of these more sexual populations. So the natural thing to do is then to go back to the experiments and have experiments in sexual populations of budding yeast. We can do haploid or diploid populations. And basically, this slide is intended for yeast geneticists, really, not for this audience. The point is that you find a lot of different ways to kill the yeast if they don't have sex exactly when you tell them to. Okay, and this was, you know, some complicated strain construction done by a postdoc in my lab, Mike McDonald. And the upshot of all this, just very quickly give you a sense. Sexual populations evolve more rapidly than asexual populations, okay? Presumably because they don't have as much of this interference effects, okay? And beneficial mutations can more easily just fix, even if they occur on bad genetic backgrounds. Whereas in the asexual populations, many of the beneficial mutations go extinct because they happen to occur in not as good genetic backgrounds. And if you look at the same kind of dynamics that I described for the asexual populations, I'm sorry, this still looks crappy because this is still ongoing, but this is sort of some preliminary data from a couple of populations. This is an asexual haploid population, sexual haploid population on the top, and asexual haploid population on the bottom. It's the same thing, you know, each of these lines is a mutation that occurred. The colors are arbitrary because we haven't we haven't annotated everything in this figure yet. But you can sort of see immediately the dramatic difference between these two. This looks kind of like those figures I was showing you from before, where you have many mutations go extinct and others fix in clusters. Whereas here, everything appears to be much more independent of each other. And you just have all this stuff going on at once. Here's a little bit of a zoomed out view of a few more of these populations. And so, you know, one of the things that we're doing now is characterizing in detail the difference between these two sorts of things.
When you say sexual, so it means that you let them mate, disorderate, and mate again, or do you try to control the number of events of mating?
We do, yeah, we control the number of events, basically. And so we sporulate, we grow mitotically for a little while, we sporulate again, etc. And we just repeat this process, and by tuning the time in between, you can effectively change the effective recombination rate.
Because in wild yeast, they always cycle between different ways, so maybe even more complicated.
It's a little bit of an open question in the wild how often yeast actually outcross. I think that there's some like controversy over it. Actually, I think many people think it's actually very, very rare, and that wild yeast basically evolve almost entirely asexually and actually outcross only once every 1,000 generations, I think is a typical estimate. But that's still an open question. It's not really clear. But here we're just using it to tune the thing to look at these dynamics. All right, I'm going to stop there because I'm out of time. I just want to acknowledge a bunch of folks. So all of this theory work has been done in collaboration, particularly with Alex and also with Josh, Richard, and Oscar. All the experiments are done in collaboration with Greg Lang. These are 4 of the graduate students who have done a lot of the theoretical work that I told you about, as well as here are a bunch of graduate students and several postdocs who have done some of the theory, and particularly a lot of the experiments, particularly Dan, Sergey, Mike designing these sexual lines. Yeah, those are the 3 folks who I've told you the most about their experiments. So I'll stop there.
Anybody final question or two?
To what extent do you expect the picture to change because of the dominance or recessiveness of the mutation?
Yeah, I mean, quite a bit actually. It's a really interesting thing. I mean, we don't really know yet. I mean, that's one of the things that we're spending a lot of time doing now is trying to look at diploid populations and carefully compare them. Here are a couple of preliminary examples. So here are sexual diploids and asexual diploids. And you can see, for example, these 3 mutations are actually over-dominant, as it turns out. And so that is presumably leading them to sort of maintain at 50%, even though these are sexual populations and they could easily fix. You know, here, I believe one of these is over-dominant and others are just dominant, but this is an asexual population, so they just can't all fix. Obviously, all these dominance effects are going to have a big impact. And presumably, the mutational spectrum will be quite different as well, because, you know, the mutations that are beneficial in haploids, you know, many of them may just not be beneficial in diploids. Certainly, there appear to be a lot less mutations in diploids than in the haploids. Part of that may be an ascertainment issue, which we're trying to sort out right now, but I think there may be part of that is real just because there are less beneficial mutations because of dominance.
This question behind somewhere here.
I just want to ask so you sampled that you had 24 clones, and I think, do you have the data sets? Are they available to see what the clones at the end look like? Maybe you did some samples in the middle so that you can.
Yeah, so we have a bunch of these things. None of this is published yet, but yeah, I mean we have all those sequences for sure. And yeah, so you can see through there. And I mean that it's useful. I mean we've used that a lot also just to sort of, you know, confirm things that we've inferred from the time courses, or like, you know, Thierry was asking a while ago about making sure that we had the haplotypes right and so on. So we've used it for that kind of thing, but I mean we have all that data, so I mean, if anybody's interested, you can email me, but it's not published yet.